I am Simone Sprio. I belong to the Institute of Science and Technology for Ceramics of the National Research Council. Our research group counts 13 persons and is particularly dedicated to the development of implants for regeneration of tissues, in particular regeneration of bone and cartilage in articular regions such as the knee or the elbow, and the regeneration of bone in spinal regions such as vertebrae, particularly in case of degenerative diseases such as osteoarthritis and osteoporosis. In particular, we develop biomimetic implants able to guide cells for regeneration of well-organized bone tissue so that the new bones can exhibit the same functionality of the existing bone before the disease. This is very important to recover the functionality of articular regions and to allow patient to stand in upright position and to recover his ability to walk and to move. So I describe a little bit our materials. On the left, there are hybrid composites able to reproduce the structure and composition of bone because they are synthesized by a process similar to what occurred in the human body during the formation of natural bone. In particular, we allow the fibration of collagen and the contemporary nucleation of nanocytes apatite. Apatite is a calcium phosphate, which is the mineral part of our bones. And in this way, we can obtain a material that reproduce the composition and structure of bone. We can also prepare materials that exhibit graded mineralization so that we can mimic the bone, the mineralized cartilage, and the hyaline cartilage of complex articular regions so that we can regenerate in one step all the tissues in that region. An improvement of these materials is the mineralization of apatite that exhibit intrinsic magnetization. This is a very new and recent advance in our research, and we foresee that this new material can help the regeneration by improving the development of angiogenesis inside the implants. The big problem in this case is that in case of regeneration of extended bone portions, often angiogenesis is insufficient to wet all the part of the implants. By aid of external magnetic fields, we can drive cells and also growth factor to move inside the scaffold, inside the implant, and to drive angiogenesis well inside the implant, so that this can help the regeneration of the whole bone missing. On the right, there are described new bone cements that can be used for regeneration of vertebral bodies collapsed due to osteoporosis. So far, the most used material is an acrylic cement that can soon harden and sustain the vertebra, but it has several drawbacks that can limit their use. This new material is a bioactive and bioresorbable bone cement that beyond sustaining the vertebral body after injection, it can induce the regeneration of the new bone differently from what occurred from the acrylic cement, so that the patient can recover the functionality of the spine, and this is very important for walking and to have a normal life.